Thanks for inviting me to this workshop. I am going to talk about host residual DNA analysis methods. Why? Because it's one of the release tests for a drug, and there are regulatory requirements for allowable limit or recommended limit of DNA in the drug. In biologics, we are talking about injectable drugs, and nobody is interested in getting injection of host DNA. So today, we're going to discuss the method, the improved methods, how we use the Bio-Rad's Droplet digital PCR technology, and how we change the testing paradigm. So this is an overview of drug purification production process, where cell line is developed, then you do a fermentation, and the output goes into several purification steps. So all these require, or specifically the purification steps, require analytics to see how good the purification process is and how the purity is advancing with every step. So analysis of process intermediates, so intermediates coming out of all these steps, and the final drug substance supports purification process development. Now, process-related impurities come mostly from the host cell, like the host cell protein, host cell DNA, and also from the column, the purification process. In most monoclonal antibody drugs are purified with the protein A column, and some of the protein A in the column leaches out and has to be monitored in the drug substance. So these impurities pose safety concerns, and specifically for residual DNA, the recommended level is 10 nanogram of DNA per daily dose of drug. Now, the current testing paradigm is this, and I could have included here in this diagram the previous. For example, it used to be dot blot hybridization to quantify or detect the amount of DNA in the drug, and then later there was a immuno-based threshold methods, and later PCR or qPCR was employed for quantification of DNA. But all those methods, the paradigm is you have a sample, you extract the DNA, and then the test method is hybridization, immunologic detection of the DNA, or qPCR amplification of the DNA for quantification. Now, there are many methods of extraction. The PCR can be probe-based or dye-based, but no matter how this step is sensitive or efficient, the DNA extraction step kind of limits that efficiency. We are kind of dependent on the extraction efficiency. So the paradigm we wanted was this. We want to eliminate the extraction step and be able to test the sample directly. Now, these samples, as I said, are crude or in process or drug substance. Has various amount of DNA, but various amounts of protein also. And is it possible to do this? Now, we had a long way to, in developing these methods, come to this stage, but it is possible, and we here showing a few examples of what we call direct method, where we are testing directly the in-process or drug substance in PCR, bypassing the DNA extraction step. So here, we use drugs made in yeast cells called Pichia cells or E. coli and put them directly in Bio-Rad's Droplet digital PCR system or qPCR system. And it worked. So you probably the next slide will show, yes, the streamline. Now, we have a sample. We put the samples in a 96-well PCR plate. We add this Bio-Rad's super mix. Now, this was a special super mix where the background level of DNA, because the super mixes are DNA polymerase and other substances, and some of the substances are made in E. coli or Pichia, so they have their own background DNA. So Bio-Rad made this special super mix where the background DNA is very low. So you add either spike with DNA to measure the recovery or the accuracy of the method, primer probe, seal the plate, and put it in this machine, which is a droplet generator. So in brief, the droplet digital PCR is basically, if you think about a PCR reaction in a 96-well plate, in one well, various PCR reactions are going on. And if you think about some of the reactions with some target is highly efficient, some are low efficiency, what your readout is an aggregate of that. Now you think about partitioning that reaction into droplets, say 20,000 droplets, 20 microliter into 20,000 droplets. And now the reactions are happening inside of each of these droplets. And at the end of the reaction, you don't monitor the progress of the reaction during the PCR. At the end, you look at the droplets. And you have put some fluorescence dye or probe that fluoresces with PCR. And you line up the droplets, and you are just looking at, is it fluorescent or not, and how fluorescent is it? And based on that, you get your data. So basically, that's what droplet digital PCR is. And that is done with this machine, the droplet reader, and there is a software to give us the data. And this is what one kind of data representation is here. Here, the blue droplets are the positive droplets. And this is, so B03 is one well subdivided or apportioned into, say, about 15,000 droplets. These are the negative droplets based on the fluorescence amplitude. And based on this threshold, which the application can draw it or it can be drawn manually. And this slide also shows a serially diluted standard. And the water means there is a NTC, no template there. So you get a serially reduced number of positive droplets. and increased negative droplets. The determination of your target DNA is based on this distribution of positive and negative droplets, irrespective of standard. So this slide shows standards alone, one drug. This is a FC fragment of a antibody directly added to the reaction mix, and the same serially diluted standards. And the last one is insulin, and this is for Pichia host, and this insulin was made in Pichia cells. Here, I believe this is 5 milligram per ml drug, and this is 10 milligram per ml drug. There is a quenching of the signal, but that doesn't affect our determination of the host residual DNA. And that's an advantage of digital PCR over qPCR. If you think of this reaction in a qPCR, the M plots would be very low and flat. But in case of digital PCR, we just need to know how many positives, how many negatives above and below the threshold. This slide shows E. coli digital comparison of digital PCR and qPCR. Here, this part is qPCR. We spiked these 3 levels of DNA with the drug. We diluted the drug to 3 milligram per ml or 1 milligram per ml. At 5,000 femtogram spike, the recovery is 100% or 95% with 1 milligram per ml drug, but less with 3 milligram per ml. The same drug, when tested with digital PCR, no matter how the drug concentration was, 1 milligram, 3 milligram, or even 5 milligram, the recovery was close to 100%. So we show that in digital PCR, the drug tolerance is better. We could test higher amount of drug, basically increasing the sensitivity of the method. So that's another advantage of digital PCR over qPCR. Now, this is the summary showing that the range is pretty big enough for our good analytical method. It's sensitive, can be used widely with these drugs, and once the method is quantified, there is no need to run DNA standard. So anybody who runs qPCR or residual DNA analysis test, the one headache is to keep the DNA standard, which is a essential thing, and you have to store it and test it from time to time. With using digital PCR, you no longer need to do that. However, the direct method doesn't work with monoclonal antibody. So we have tested interferons, insulin, modified insulin, FC fragments with E. coli and Pichia host because that's where they are made. When you test, say, monoclonal antibody made in CHO cells, it doesn't work because monoclonal antibody, if you put a monoclonal antibody through the PCR thermocycle, heating at 94 and then cooling at 70 or 60 and heating again for 40 cycles, it coagulates. You get a gel. There is no light that crosses that gel, and the reading is nothing. You found nothing. So, but we were intent to changing the paradigm of DNA extraction. We have been looking for a protease that will digest the protein, because we are interested in this protein mix, how much DNA is there. So we want to digest the DNA. If the protein is the obstacle, let's digest the protein. The DNA will be spared. We are interested only in the DNA, and we'll amplify the DNA and quantitate it. So that's the idea. We found a protease that probably was derived from proteinase K, but engineered, proprietary, we don't know much about it. Proteinase K, we have tried proteinase K. Optimal digestion temperature is about 60, close to 70. But it doesn't denature well at 90 or 95 degrees. There is a remnant, and that's enough to kill the DNA polymerase. This protease is operational at 60 C and can be denatured completely by incubating at 90 C for about 10 minutes. 5 minutes is also enough. So we developed this method in a qPCR setting where you get the sample, we have the kappa enzyme, that's the protease. We add other things, we cover it, do a protease digestion, and then denaturation of the protease. We remove the cover and add PCR reagents here, put a transparent cover back for PCR, and run the PCR. Now, what we noticed was, we immediately noticed that the recovery or the accuracy of the method improved a lot. DNA extraction-based methods, we have been struggling with recovery. 50% recovery, we'll be happy. But now we find that we can get 100% recovery. And the other thing is, we have eliminated one instrument, the extractor, from the equation here. It's less time, inexpensive, it's easy. We can, about equivalent amount of drug we can test. But the accuracy is close to 100%. And this is truly 100% because it falls on both sides of 100. And sensitivity meets requirement. What we have done is next, we tried this method in a digital PCR, but our probes, primer probes, really didn't work. So at that point, Bio-Rad stepped in and they said, we have this primer probe for CHO host residual DNA, you try it. So they supplied it to us before they marketed it, and we used it in the same method by kappa digestion and then making droplet and running the PCR. And this is the graphic version of the method. We have test sample, we put it in a 96 plate, we do the kappa digestion, then we open the cover and add the droplet digital PCR reagents now. We make droplets, we put in a plate, run PCR. This can be any PCR because it's an endpoint determination. And then read the positive or negative droplets. And we have successfully carried out this and completed a droplet digital PCR method. This method is still not in our regular use, but the E. coli and the PKM method we have transferred to GMP lab, and soon will be used in our probably commercial products. So this is a comparison of all these different, mostly monoclonal antibodies. This is the amount of drugs tested. And digital PCR and qPCR data is more or less the same. A slight difference here, a little bit more different here. And that data is based on no detection. There is a detection but below LOQ and calculated based on that. But for digital PCR, we had a number and we could, that was above LOQ, and we could calculate the exact amount of DNA. So now the question is, if we go back to that model of DNA extraction and testing, and our desired model of not extracting DNA and testing the sample, we have solved the CHO monoclonal antibody testing by adding protease and digesting the protease. But the protease, what it is doing is, if we subject it to thermocycling, it's precipitating. But if we don't subject it to thermocycling, and the protein remains in solution, we are able to target the DNA, and somehow, if we can amplify the DNA, we have a method, a truly direct method without doing protease digestion. And that's what we are trying now. Most of what I showed in the previous slides are published. This is not published, and we are in development. And we are doing it in a qPCR setup. Now the question is, will we be able to do it in a digital droplet PCR? The problem here is the droplets generated, this is an oil and water mixture, and we are able to put it through thermocycling because the droplets are stabilized by baking. In the beginning of the process, we have to bake the milky droplets in a PCR well at 95 degrees for a few minutes. Now the droplets are stable, so the phases don't get mixed up. Here we are talking about a lower temperature where the protein does not precipitate. Unfortunately, the droplets are not stable. So at the end of, if we were to do this, I've done this, at the end of the process, we'll get two phases, water and oil. Droplets are made, but if we subject it through thermocycling, what it's doing is, I think the PCR reactions are going, but it's separating into two phases, which is no good for us. The whole pain of making all these droplets and trying to figure out whether the droplets are positive or negative doesn't work. So if Bio-Rad used to develop a oil that will stabilize at, say, 60 degrees centigrade, then we will be able to do it. With qPCR, we are able to do a thermocycle, isothermal PCR. We are able to amplify the DNA in the drug mixture. So this shows a typical amplification plot of standards alone and standards added to the drug, and a little bit of the drug added to the qPCR and subjected to this isothermal PCR, and we are able to generate this kind of amplification plots. So hopefully we'll be able to develop a method where a simple analytical method for host residual DNA will be available. I think, so this is the conclusion. We have changed the paradigm, which is a simple thing to do, that let's not extract the DNA and be able to test directly. We have used digital PCR, and basically that's it. I say thank you to Bio-Rad for its support and other members of our lab. Thank you very much for your attention.